This is the Meso Scale Discovery S600 sector imager, also known as MSD for short. It's an electrochemiluminescence platform. It's another readout for a plate-based ligand binding assay. So what that means is if you're doing early candidate selection for antibodies for your assay, great, you can pair them up capture detection. If you're working in preclinical space, if you're working in the clinical space, these machines support everything from the early discovery work to phase 3 sample analysis and going to new drug approval. One thing that's unique about the MSD is that it's a format that allows for a very easy bridging assay. So what that means is in the context of immunogenicity, we're developing an anti-drug antibody assay. We're trying to understand if the patient is producing immune response that binds to the drug. We establish that by having differential labels on it. So if the antibody is multivalent or bivalent, binds to a capturing antibody, binds to a detection antibody, signal has a relationship with a dose response. Dynamic range is the big one. They'll tell you on the catalog that you're going to get about 6 log dynamic range. In practice, it's not far off if you have well-characterized reagents. It's also extraordinarily consistent. There's no enzymatic steps, so when you are getting your assay readout, there's no art in understanding of what your radiosignal development is. It is add reagent, acquire data, and for that reason, it can be very consistent over time, over technique as well. The machine's importance in one sentence: it has a very healthy level of support for consistent quality from both a manufacturer standpoint as well as data validation. So it's consistent. As for an example for where this has impacted a project, I've got to work on some immunogenicity assays that are for atypical mutagens. In this case, I'm thinking of an assay where it was actually for a siRNA payload. So not a protein, but there's still be an immune response against it. Those are typically very low affinity, very weak, and they're poorly characterized in literature. So you need a very sensitive platform to acquire that. And MSD was the one that got over the finish line.